Hello, we're glad you joined us for this webinar, Improving Lipidomics Performance, a View Through a Local Lens. I am Michelle Ashton of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, a leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration. It's sponsored by Agilent. To learn more about our sponsor, please visit www.agilent.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation via email. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Ask a Question box located on the far left of your screen. I now present today's Bruce Kristal, the Associate Professor at Brigham and Women's Hospital in Boston, Massachusetts. To learn more about our speaker, please visit the Speaker tab at the top of the presentation window. Bruce will now begin his presentation.
All right, I'd like to thank you today for listening to a presentation on improving lipidomics performance and a view through a local lens. I'll start with my disclosure. There's no specific issues that are scientifically related to what I'm going to talk about. I have in the past licensed 10 patents to Metabolon, all of Metabolomics. I have consulted for them in the past. I hold non-public equity, and I do get post-sales royalty, everything of which was through a prior university. And I have no other talk-relevant affiliations in the last 5 years. I want to start with this, and this is really the central message that I want to talk about, which is that lipidomics technology is a lipidomics platform. It's not simply one component of the system, for example, the mass spec, but it's everything put together. In particular, in the context of what I'm going to talk about, it's the triad between the biological system, the analytical system, and the data analysis system. Now, in today's talk, I'm going to focus really only on the analytical chemistry side, and I'm going to talk about it conceptually. I'm not a mass spec person. I'm not a chemist. I'm a biologist. But what I want to get across are the conceptual advances that have been made and how these move the field forward. And again, I want to talk about the fact that this is a view through a local lens. And what I mean by that is this is a convenience sample. These are a set of experiments, a set of papers that come out of my lab over roughly the last 7 or 8 years. And they are therefore convenient for me to talk about. But it goes beyond that. So first of all, these are the papers. I'll refer to them throughout the talk. You'll have the list. They'll all be highlighted where it's relevant. But the key point here is that this is conceptually biased by my interests. These advances, these papers, these studies were all based around the fact that we're trying to answer questions in biology. And by no means should you think that we're the only lab that has advanced lipidomics, or by any stretch of the imagination that I think that. That really what you're looking at are the changes that we've made to solve problems. And you could take multiple other lipids labs across the world that have done similar things. They've, for example, increased quantitative accuracy, or they've increased precision, or they've increased the ability to see certain lipids, or to see those lipids faster or more sensitive. In all cases, they're solving problems. And it's important when you're looking at lipidomics, particularly when you're looking at lipidomics as a field or a lipidomics paper or a lipidomics study, to realize that in a lot of cases, you're solving specific problems. And another way of saying this is that lipidomics is by no means a fully mature field. It's not a single assay. It's not really a set of assays. Okay, it's a lot more than what often gets viewed from the outside, that it's simply a matter of how cheaply you can do it or how fast you can do it. Now, there are cases where cost is the dominant factor or where speed is the dominant factor. But there are also cases where it's depth of search that's accurate, or making sure you don't have false positives or don't have false identifications. And so again, it's a fit-for-purpose assay, and it's really important to keep that in mind. And analytical chemistry is not a science. That surprisingly gets lost and taken for granted in a lot of the discussions I see in conversations, for example, with clinicians or with biologists, who lose the fact that this is a rapidly evolving scientific field. And arguably moving it forward in many ways is still an art, as we don't know exactly the best way to improve what we're looking at. So our central question for well over 20 years now has been a metabolic serotype of diet, lifestyle, and environment. In other words, developing a way to look at a person's blood or other biological fluids and identify markers for disease risk. Not so much for diagnostics, but can we figure out what disease this person is at risk for? And you can conceptually embed this in the concept of how do we study complex biological or clinical research problems? Or you could say it a different way. How do we address such questions in humans where our ability to manipulate and analyze the system is very limited? And that goes back to this triad. You have to think about the biological systems that enable you to address this study. You have to think about the analytical systems that will enable you to get the data that you want, and the data analysis approaches, the informatics approaches that will give you the insights you're looking for. In our case, where we're looking at a very large project, and we're looking at a project with very low signal to noise, and we're looking at, by definition, subclinical phenotypes, we don't know what we need. We need to profile. Targeted methods are not going to help us. So breadth of coverage is, for example, very important for us. You can, again, conceptualize what we do in this type of a diagram. And what you're looking at on the leftmost side is sample collection, okay? Or at the bottom, the outcome, you want mechanistic insight, you want drug development insight, or classification and prediction in our case. You may want to do functional genomics, how we link phenotype to the genome, or, as I said, sub-threshold studies. This is the biological aspect: sample collection, the choice of an analytical platform, and the biological questions being asked. Analytically, you have to worry about sample collection. How do you need to do sample collection to get the studies you want? Again, what's the analytical platform and exactly how that analytical platform is developed? And then you need to do the data reduction. That's an analytical chemistry problem, to decide what data should be in that database. Then you have a data analysis component. It begins with understanding the analytical chemistry platform. You have to know what your platform does and doesn't do, and doesn't do well strongly. You have to clean up that data, and you have to understand how that cleanup works and what its strengths and weaknesses are. And then you have to choose the appropriate bioinformatics tools. So if we now switch and we focus on lipids, you can broadly break lipids into 8 classes: the fatty acids or fatty acyls themselves, the glycerolipids, the glyceryl phospholipids, the sphingolipids, the sterols, the prenols, the polyketides, and the saccharides. In our case, where we started this was an involvement in questions along mitochondrial lipids, mitochondrial biology. And we started by building off a platform that had originally been developed by Hankemeyer. It uses a high-resolution, high-mass spec platform. And I'm not an analytical chemist. I won't go into the details of what I'm talking about. If you want the details, go to the papers, because the papers will have those details. But basically, we run both a negative and a positive ion. It's an electrospray instrument. And we're looking at the fatty acyls, the lysoglyceryl phospholipids, the glyceryl phospholipids, the cardiolipins, the sphingolipids, including the gangliosides. We're looking, again, in the positive ion mode, the lysophospholipids, the glycerolipids, including the mono, di, and triacylglycerides, the acylcarnitines, the sphingolipids, the sphingomyelins and the ceramides, the prenols. And we get a lot of information. If, for example, we get over 100 phosphocholines out of the mitochondria, we get over 60 phosphoethanolamines out of the mitochondria. 31 phosphoglycerols, and so on, all the way down. A lot of ceramides, a lot of triacylglycerols that are the structural lipids in the mitochondrial membrane. The triacylglycerols. We get a lot of ceramides. We get the sphingomyelins. We get the gangliosides. And we get the triacylglycerols within the mitochondrial fraction. The glycerophospholipids, the monoacylglycerols, the diacylglycerols, the phosphocholine, the plasminogen phosphocholine, the lysophosphocholine, the phosphoethanolamines, the plasminogen phosphoethanolamines, the lysophosphoethanolamines, phosphoglycerols, phosphoserines, phosphoinositols, and the cardiolipins. At the time the platform was developed, we can see a lipid that's present at a couple of lipids per 100,000, with about a 5% CV at a lipid that's present roughly at 1 lipid in 10,000 pieces. Mass accuracy is about 2 ppm. And in this untargeted method, we can already pick up more cardiolipins than we see in the targeted method. So in essence, what this is doing is it's turning the non-targeted method into a semi-targeted method. In other words, it's blurring the lines between the non-targeted and the semi-targeted method. And again, if we go back, we can take this same platform and we can now apply it to a serum pool. And now we see the serum triglycerides. And these are basically the first 2 papers published. And I'll point out that these 2 papers were, first author was Susan Bird, who did about a lot of the analytical chemistry and the lipids in the lab. And the other person who does a lot of it is Rose Gathungu. And they were the 2 people who ran the lipids platform in my lab during the time of this. So now I want to switch gears and I want to talk about biologically what we were doing. And here in this study, we were focused on looking at the interaction between mitochondria, macronutrients, and the blood metabolome. And I'll stress here it was the blood metabolome initially. But what I want to talk about is about bottlenecks and leverage and the ability of a lipidomics platform to get you through these. When we started this, if you look at this interaction figure, very little was known about the interaction between diet and mitochondria, but a tremendous amount was known about the other interactions within this figure. So we hypothesized that diet-associated effects on mitochondria are linked to diet-associated changes in disease risk. And I'll point out this was a very complex study. It was looking at developing biomarkers out of serum. It was looking at developing target validation. It was looking at biomarker outcome validation. And the study itself was fairly complex. We had a 24 dietary group study in animals. The animals were fed ad libitum for 8 weeks. We then were measuring food consumption and growth in the animals. We had multiple practical functional assays in the mitochondria, all of which were then taken in through our scripts for the informatics. And we had a lot of biochemistry that was planned, particularly on the stored plasma samples. When we did the study, however, and this is a mitochondrial functional study where the upper left is looking at, in essence, the idling state of the mitochondria, the upper right at the functional energy production state, the lower left at the ability to switch between these states, and the lower right at the ability to couple these states tightly without losing energy or generating free radicals. There are well over 16,000 comparisons in this slide, and we had less than chance that we're statistically significant. So we were really at a major bottleneck because this was, in essence, how the entire grant was designed. So this is a gigantic bottleneck. And because of this, we switched to what we added in was a mitochondrial lipids component. And if you go back to this triad, we have the rats, the biological system, the biological questions, or mitochondrial function, for example. We added a lipidomics component, and we added some data analysis. And what this does is it allows us to leverage what we're doing. So we now add in biochemical mitochondrial targets, which were not originally a plan. And we start with the idea of looking at cardiolipin. Cardiolipin is about 20% of the phospholipids in the mitochondria. They function as a proton trap. They're essential for mitochondrial structure. They're essential for mitochondrial enzymatic activity. When you have mitochondrial dysfunction, their concentration decreases, the acyl chain composition changes, and there's oxidation. And I'll point out that this is arguably the central lipid in energy metabolism. Okay, there's one other lipid that could compete for this, and we'll come back to that a little bit later. But we see 28 structural variants on the cardiolipids. And as I pointed out earlier, at the time this was done, that was more than was seen by targeted methods. We had developed an informatics platform which enabled us to, in essence, reduce any given lipid into a series of 14 pages worth of informatics, and they could be scanned quickly. And what you see on one of the cardiolipids is two of the diets have vastly altered levels. That's the dark squares in the upper left. And when you look at this on a dot plot in the upper right, you see that these are actually saturated fat and trans fat fed with high glycemic index. So in other words, cardiolipin has actually increased in the bad diets, and it's increased a lot. But strikingly, what's more or less known in mitochondria is that cardiolipin never goes up. Okay, so this is some sort of compensation effect that's never really been seen. But then the question becomes, what's driving that? Well, we profiled. So even though we had no intention of doing it, we now said, what could cause this? Well, the obvious thing to look at is the other major lipid, which is the ubiquinones. So these are the prenol lipids, the coenzyme Q, or the ubiquinones. And there are three major ones in rat liver mitochondria. For all practical purposes, the major one is coenzyme Q9. It sits within the electron transport chain. It's the dominant electron transport lipid within the mitochondria of rats. In humans, the dominant is CoQ10. But CoQ9 goes down. What's striking there is that ubiquinones never go down in the literature without drastic losses of mitochondrial function. And as I showed you earlier, there are no changes. So what's going on? Can we look at oxidative stress in the lipid component? And ideally, we want something that might accumulate. And again, we can take advantage of the fact that we profiled. So we can go back and look for things that we never thought of. In particular, we look at what are called monolysocardiolipins. These are cardiolipins that only have 3 fatty acyl chains. And that could be because one has been damaged and knocked off, or it could be during the synthesis phase of the cardiolipins. And we're fortunate that we see 2 of them. They are minimally abundant. They're present at a couple of copies per 100,000 lipids. And strikingly, they are also increased in these 2 diets. So yes, we can see something that might be accumulating. Can we look at something that might be much more clearly oxidative stress derived? And what had been shown by another group was that 18-1, 18-1 lipids undergo cis-trans isomerization in the presence of thiol radicals. Okay, so for example, if you look at the cis-trans lipids, okay, what you're seeing is normally you have a cis shape, and it makes the membranes more fluid. Okay, but when you oxidize them, you hit them with a thiol radical, and they undergo a set of reactions, they now give you a trans lipid. And these trans lipids turn out to be separable in our system on a series of C18 columns. And in fact, we can detect these 2 molecules differently in a pooled mitochondrial sample. And this method has entirely been published. And what we see is, again, the cardiolipins are this cis-trans isomerization is increased in the case where you have these elevated cardiolipins, and we believe you have evidence of oxidative stress. We now can go back again because we profiled, and we can look at the entire cardiolipin pool. Cardiolipins typically are believed to be the backbone and 4-18-2s. We do know that there's some variation in this. When we look at the variation, in fact, we see that there are enormous changes that we see. For example, this one lipid is extremely elevated in animals fed saturated fat diets. Okay, so there's a level of regulation that really wasn't anticipated. When we look at this in more detail, if you just focus on the bottom center panel, what you see is that the predicted, based solely on fatty acids randomly shuffled and reassigned, permuted. One of them has a much lower level than what's calculated. So the observed is the top, the calculated is the bottom. That's the red arrow. And 10 of them actually have higher, in some cases, much, much higher levels than would be anticipated. So there's both positive and negative selection for which variants of cardiolipin get kept into the system. So this, to some extent, this overall study has altered the concept of ubiquinol respiration regulation and evidence for as many as 5 new levels of cardiolipin regulation. And again, this part of the talk is about bottlenecks and leverage. You see what these more advanced lipidomics techniques allow you to do. It took about a week of mass spec time. It bypassed the bottleneck. It quadrupled the actionable information from a $1.7 million study, and it allowed us to leverage all of the other data for this. And again, all of these work has been published. So let's switch now from the mitochondrial side of this to a completely different matrix. So we started to look at fecal lipidomics. In particular, we did it because it offers a biological and analytical advantage for the study of neonatal GI disease. We know that lipids are central to intestinal biology. They're more stable to many metabolites, and they're more conserved than proteins. The use of fecal matter has huge advantages in the sense that it's a completely non-invasive approach that doesn't deplete the premature infant of its limited blood volume. So you get the same basic system, except you've replaced those top panels with the infant, fecal lipid extraction, and a different analytical platform. And what we focused on was method development. We start with a pooled sample. We look at a dilution series. We look at different extraction buffers, and we looked at it with and without the presence of pressure. Overall, it's a 60 sample study. And we see that as we space out across the chromatography, you see, first of all, different lipids that are better extracted in MBTE versus dichloromethane. We see lipids that are better seen in the absence of pressure. We see ones in a second I'll show you that are better with pressure. And we see the analytical criteria. We see whether or not we're getting a 3-to-1 difference in dilution. So we can see how effective the dilutions are. And what we see, and this is obviously in a lot more detail in the paper, is that everything makes a difference. In some cases, the dilutions do not work. You can have far too much. You can have too much material for the extraction buffers. In some cases, pressure helps. In some cases, pressure appears to create artifacts. You get more lipids with some scenarios. You get less lipids with some scenarios. Sometimes you get more lipids, but they're more error prone. So again, this becomes a very fit-for-purpose type of analytical system. But it allows you to look at this biological system and ask questions that are going to be relevant. And again, this work has been published. Now what I'd like to do is to switch systems again and to talk about a study that we did ancillary to the calorie trial. So the aim of the lipids component of this was to develop, optimize, and validate a defined series of nested plasma lipidomics-based biomarker profiles. And it builds off work that we had done in the rats looking at rat markers for caloric restriction. And we have, in the time we did this analysis for the first time, we had 504 structurally identified plasma lipids. About 200, almost 200 of them were triacylglycerols, almost 100 of them were phosphocholine. And what we see in this particular case, looking at the triglycerides, was that we really didn't see separation based on resting metabolic rate, resting on total energy expenditure, or looking at the groups. In other words, ad lib versus CR as they were defined in the trial. We didn't see it in males. We didn't see it in females. It was a very negative result. But we know in the rats that even a 10% CR in the female brown Norway rat, and I will point out for a second before I forget, this entire study is not published, although it's all been presented multiple times. So you would expect to see this. So in the humans, in contrast, we seem to see minimal effects, or not specifically More specifically, nothing really replicates compared to what we would have expected for the rats. And to a certain level, this defies what we would think of as common knowledge. We know that people who lose weight tend to have lower triglyceride levels, and we don't see that, okay? So even though we didn't get to 25%, which is what the trial had hoped to do, the 10% or 11% or 12% should have been more than enough to see this kind of effect. So maybe it's just a triglyceride effect. And in fact, we tested this, and this is not true. So one of the things we looked at was what's called hot box. It's a commonly used method where one heats an arm before a blood draw. It's used for different things. In particular, in this particular case, it was used for catecholamine draws. It was used as the baseline, as part of the baseline 12-month and 24-month work. Those were the first tubes drawn. We got one shortly after it. And we wondered if this could make a mistake, could be causing issues. It didn't have an effect at the population level. There were no obvious increases or decreases in triglycerides, but maybe there's some sort of pooling issue. When we looked at the energy lipid associations at 0, 12, and 24 versus 6 and 18, we see that those relationships are really no longer related. So the take-home message here is that sample collection matters in really unexpected ways. If heparinized tubes are allowed to sit around for a long time, you see an alteration in the lipids. There's a lipase activity. And so this needs to be considered when you're dealing with sample collection, as a fit-for-purpose aspect. So now I'd like to change gears again and talk about reducing misannotations. So we were originally part of a ring trial in lipids. And when we started to do some of that work, we got a couple of results that didn't make sense to us. So we stepped back for a little bit to figure out what was going on. And what Rose Gathunga, who was the lead on this, showed eventually was that if you have, for example, lysophosphocholine, you can get in-source fragmentation. So this is fragmentation within the electrospray source that actually leads to biologically valid, in other words, biologically real lipids that weren't in the initial sample or were at different levels in the initial sample. In the particular case shown, the in-source fragmentation of a lysophosphocholine 16O can give you a lysophosphoethanolamine 18O and a fatty acid 16O. By taking advantage of the chromatography, and again, the fact this is a profiling method, we can see that the fatty acid 18-1 runs in two places if we look at an extracted ion chromatogram. One of them, in fact, the dominant one, is actually a fragment of the LPCs. It's not a real free fatty acids. Rose determined that you can see this in multiple cases. For instance, from the LPCs, you can see it from the LPEs, you can see it from the PCs, you can see it from the sphingomyelins. And when we actually looked, focusing in this particular slide on the data that is actually in the NIST data set, and we worked with John Bowden and Jeremy Cornell on this, what you see is that about 10 of the lysophosphoethanolamines are potentially fragments. They're potentially not real in the database. And what was done in silico was to basically determine all of the cases where this can occur and to make that available as a data set. And again, this is all published. Finally, I want to turn to improving identification. So we talked about reducing misannotations, misidentifications. One of the issues is improving phosphoserine detection. So phosphoserine is a little bit harder to detect. It also is typically lower abundance in biological systems. And Rose took advantage of the fact that deuterium labels phosphocholine. So if you look at the inner and the left and right lipids here, the phosphocholine is on the left, the phosphoserine is on the right. They have the same mass. But if you expose them to deuterium and allow hydrogen exchange to occur, you get the deuterated salt of the phosphocholine. When you now run this, if you start in the normal buffers, you have two lipids with identical masses and identical isotope patterns that are indistinguishable. Whereas if you add the deuterium, you can now see that it's the lipid that's running, in this case 1321, that's the phosphocholine. And again, this method has been published. So what are the challenges going forward? If you go back to the global question, biologically, what are the design questions? Valid controls, statistical power, determining what the hypotheses are, determining what the confounds are. Sample collection, getting matched samples. Analytical issues I'll come back to in a second. Biological issues such as maximally appropriate coverage for the problems of interest. IDs, dealing with flux if you need to, deal with appropriate conditions under the biological systems. Determining whether or not things are robust across time with interclass correlation coefficients. And getting robust data collection and analysis. At the informatics level, statistics versus informatics, multiple comparison problem, building robust models, establishing model validity. If we go back to the analytical piece, lipids is by no means a single assay. It's by no means a set of assays. Okay, there's a lot to still be developed. There's a lot to be gained by tailoring the platform to the problems of interest. There's issues about sensitivity. There's issues about specificity. There's issues, as I showed you, about both increasing correct IDs and decreasing false IDs. There's critical questions about improving the linear quantitation range, about improving the breadth of coverage, reducing the coefficients of variation, identifying double bonds, identifying cis-trans bonds, identifying oxidative coverage. It's a wide open area that still needs development. In acknowledgement, Susan Bird and Rose Guthunger did the vast majority of the analytical chemistry. Fatima Tausi was in the lab for a little while, did some of it. Irina Stavrovskaya was the dominant biologist in my laboratory for over 15 years. She's now at Columbia. All of the mitochondrial work you saw was her. A lot of the animal work you saw was her. Matt Sniatinsky and Vasant Marur are the two informatics people who've been with me for over 10 years. John Bowden, Jeremy Colmel, who were at NIST when this was done. Pamela Stark-Reed and Van Hubbard were biological collaborators that were at NIH. Kate Gregory is a PhD nurse, originally at BC, now at the Brigham, who ran all the neonatal work and who conceived a lot of the neonatal work with Alan Walker, who's at MGH. Alex Lazarov, Vera Gross were at a company that we collaborated with for some of the pressure studies. Pablo Lara and Heather Greenberg were techs who did a lot of work in these projects. And at various points, these studies have been funded by NIA, NCI, NHLBI, NIEHS, NIDDK, and NIGMS. And I thank you very much for your time. And I'll be happy to take questions via email. Thank you.
Thank you, Bruce, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of the presentation window labeled Ask a Question and click on the Send button. Bruce will answer your questions via email after the presentation. We would like to thank our educational sponsor, Agilent, for today's educational webcast. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.